Hello, in this video we will discuss about the risk and safe pathway. What is the risk and what is the safe? Basically, the risk is the reperfusion injury salvage pathway. It is stimulated by a drug or other factors, while the safe pathway is the survivor activating factor enhancement naturally through cytokine receptor. While the remote ischemic preconditioning RIPC is temporary. So in this way, let's begin to understand first of all the introduction. What is the ischemia or hypoxia? So during the arteriosclerosis or atherosclerosis in the coronary, for example, artery, which that plaque formation will lead to inhibit the oxygen, and in this way the providing of the oxygen to the cardiomyocyte is decreased, will lead to glucose convert into the pyruvate. Is the anaerobic respiration will occur, but aerobic will not occur due to the less oxygen and lead to hypoxia and the ischemia it is known as. While on the other hand, the reperfusion after the recovery, when the some of a problem can be solved or other things can be solved, in this way the reperfusion will lead to again oxygen sudden absorption. Oxygen when sudden absorption to produce more ATP. So in this way the risk pathway stimulation is very important for the survival of the cell. For the prevention during reperfusion, acute injury of the cell can be possible due to sudden oxygen reperfusion, which can be decreased by risk pathway can be triggered by drug. So during the angioplasty or other things, when the oxygen level will be drop in the heart, in the coronary, in the cardiomyocyte, so the risk pathway will be stimulated after this function. So after this treatment of the angioplasty or other factors. So in this way here is you can see this is the cytokine receptor, remember, and this is the G coupled protein receptor. So the risk pathway involve the G coupled protein receptor, intracellular contain domain dimeric, and the extracellular contain also dimeric. So the drug when bind, this is a ligand, and ligand when bind will lead to stimulate the RAS stimulate to phosphorylation to the RAF phosphorylation to the MEC1 and 2. MEC1 and 2 is the mitogen activated protein kinase 1 and 2. On the other hand, here is the RAF is the retinoblastoma activating factor, and in this way here ARC1 and 2 is the extracellular extracellular regulated kinase 1 and 2, which that stimulate, and in this way after the phosphorylation of this protein will convert the STAT inactive to the STAT activation. And this is the reperfusion injury salvage pathway for the prevention of the mitochondrial damage. And while on the other hand, here is the phosphoinositidyl inositol 4 5 bisphosphate 3 kinase enzyme kinase protein is used to trigger the mTOR-CIV2 mammalian target of rapamycin complex for the survival and synthesis of protein, while the protein dependent kinase activate the AKT protein, this protein kinase B, and in this way this phosphorylation will lead to inhibit the sodium proton exchange channel. In this way the sodium and proton will not exchange, and in this way the proton will not move inside for increasing the acidity. So when proton when move inside will lead to acidity increase. So in this way the acidity will not increase into the cell, will lead to prevent the cell lysis or other things and plasma membrane lysis inhibition. While the AKT will also stimulate the glycogen synthase kinase 3 beta and basically nitric endothelial nitric oxide synthase 3, and on the other hand the BAD and BAX. The BAD and BAX mean the BAD is the BCL2 associated death promoter, while the BCL2 associated X protein. This is basically used for the stimulation, the triggering or inhibition of the mitochondria. Here is the mitochondria. So here is the STAT3 phosphorylation will lead to inhibit the mitochondrial permeability transition pore, mitochondrial permeability transition pore. While here is the nitric oxide will stimulate the protein kinase C, will also inhibit the mitochondrial permeability transition pore. In this way, the cytochrome will not outflux as well as other factors. In this way, the mitochondria will not damage. And here is a, you can understand, this is the mitochondria permeability transition pore, which that is inhibit by the protein kinase C and STAT3 due to the risk pathway, so the reperfusion injury salvage pathway. While on the other hand, let's begin to understand the safe pathway. Safe pathway mean it can be unnatural by the binding of the cytokines. When cytokines will bind, the cytokine receptor will recruit and docking the jack after the phosphorylation of the intracellular trimeric subunit of the cytokine receptor will docking the STAT3. After this docking the STAT3 to phosphorylation of the STAT3 will go into the mitochondria, and same mechanism to inhibit the mitochondrial permeability transition pore to inhibit the mitochondrial damage. In this way, the cytochrome will not move out. So cytochrome when not will move out, so the cell injury and cell apoptosis or necrosis will not inhibit. So in this way, the inhibition of the injured cell will be prevent through a risk pathway and the safe pathway during the hypoxia and reperfusion. So thanks for watching.